Can you see my screen?
Yes, we can.
Okay, well, thank you. Okay, so my lab is interested in understanding gene regulation during development and how that's encoded in DNA sequence. So development is a very reproducible experiment, and these patterns of gene regulation are really established in a very precise way, and that information is encoded in the DNA. And so we know in principle how this works. So we know that transcription factors bind these cis-regulatory sequences, and it's the combination of these transcription factors and perhaps also the arrangement that determines whether this region, for example, will become an active enhancer and activate genes nearby. But as you can already appreciate in this specific example, that transcription factor motifs themselves, they come kind of in different flavors, different affinities, and we don't know whether the spacing, for example, so that the syntax matters in the activation of gene regulation. And so even though this is a really well-studied problem, we really fundamentally can't read this information from sequence alone and make predictions about where these genes are expressed. Now, the good news is that we now live in an era of genomics technology that's really unprecedented and should really help in understanding this problem. So starting from DNA sequence, we can now measure on a large scale how transcription factors bind to small DNA motifs using Celix, for example. So this would be in vitro. We can also measure transcription factor binding in vivo really, really well. We can also measure chromatin accessibility. Then we can measure how enhancers become active. So when enhancers are active, they have certain histone modifications. They also have enhancer transcription, so there's a little bit of RNA involved here too. Of course, there are also reporter assays. And of course, eventually we want to look at target genes and how they're expressed nascently, or we can look at single cell resolution in RNA and also now in a spatial way. We can look at protein and we can look at perturbations. So all of this together really should give us an idea of how gene regulation works. But the problem is, how do we put all of these data sets together? So that's a very challenging problem. And I think you might agree that this can be sometimes frustrating. So here's a citation by Sidney Brenner already from 10 years ago. Technology gives us the tools to analyze organisms at all scales, but we're drowning in a sea of data and thirsting for some theoretical framework with which to understand it. And I share a little bit of that frustration. I can certainly see that. But I would argue that we do have the theoretical framework already, that it's more a technical challenge of how do we put this together in a coherent way. So I would argue the theoretical framework really is DNA sequence. And we knew this already since the 1950s. So in the 1950s, it became clear that DNA is the transforming agent. This is really what contains the heritable information. In fact, it was discovered already in the 1940s by Oswald Avery, but he wasn't believed until the 1950s. It really took a while because people believed that proteins were more important than DNA. But of course, we also know the structure of DNA from Watson and Crick in the 50s, and that became a very clear model of how information can be stored, copied, and read. And we also know from experiment in the 50s that this information can be changed. So for example, by X-ray mutations. And so this in turn changes how an organism develops. And so now, knowing all of this in the 50s and fast forward to where we are today, I would argue we're still in the same situation, where we know that lots and lots of information is encoded in the DNA. So we know this from genome-wide association studies of diseases or other traits. And so we know that the information is there, but we can't pinpoint exactly what the mutations are. And we also don't know the mechanisms by which these traits and phenotypes arise. So again, we know it contains the heritable information. We can also read this information incredibly well using sequencing on large scales, of course, We can also edit this information using CRISPR and more specialized techniques derived from there. So this should be very, very powerful, but we're really hindered by our ability to read what's encoded in the DNA. And of course, I wouldn't talk about this problem if I didn't have an answer. So why is that we can't read this information? So I would argue a big reason is that our current paradigm of doing research is to look at a few components at a time, because this is what the human mind does well. So for example, if you use genetics to put pathways together, or you do biochemistry, or you look at structures or perturbations, we can get a very fine-grained knowledge in relating this component to each other. But the problem is that gene regulation is just not solvable in this kind of approach. And you probably realized by now that neural networks, or generally sort of talked as AI, has become very powerful in our society and also in biology. So you probably heard about GPT. So these large language models are really amazing in learning very large amounts of data and generating very complex outputs. And I would say in biology, the big breakthrough has been predicting protein structure by AlphaFold, so that's done by DeepMind. And I think that has really realized that how much we can really do with neural networks. Now, I want to give you just a brief idea of how neural networks look. So specifically convolutional neural networks that are used in facial recognition, for example. So in your cell phone, it's usually very notable that it's very easy to classify people. And that's, of course, despite the fact that the pictures are, very different backgrounds and very different lighting and, glasses, et cetera, And that's because the neural networks are very powerful in learning information, even, among very, very complex data inputs and very sparse data. And the way it does that, it's trained to find rules from the bottom up. So when you give it a lot of images, it will first, so deep learning means it has lots of layers in the networks. And the first layer learns just simply what kind of structures it can find at the most lowest resolution level. So define actually high resolution, sorry. So it looks at pixels and sees, for example, edges. And now in the subsequent layers, you'll combine this information. And so it will learn that there are noses, eyes, and mouths. And then it will put this information together and eventually, so in this case, it was trained to recognize me, and it can recognize me. So what I sort of wanted to grasp for you intuitively, that we can learn, so let's say we should be able to learn motifs, sort of the basic patterns, but we're also learning how motif combinations then affects a certain output. So intuitively it should be very clear why neural networks are very well suited to do this. However, though, and usually when I talk to people, it's not quite intuitive. And the reason why it's not intuitive to biologists is that it's very different from traditional techniques. So traditionally, when we take a transcription factor binding data sets, for example, we process the data. So we're getting down to the regions of interest, and we hypothesize, for example, that there are enriched patterns such as motifs or co-enrichment of motifs. And then we're testing the statistics and finding overrepresentations. So what we're essentially getting is a very abstract representation of the patterns that we think are in the data. And we're not very confident because they're usually not present in all the genomic regions. And so then when we take individual regions, we don't really have good confidence of what's happening in that region. So and also the predictive accuracy is really low when you put your patterns together. So now we're inverting this completely. So with a deep learning framework, so I call this the inverted learning paradigm, we're training it to predict the data that we already have. So in this case, it would be the transcription factor binding data. And we're training to do it on the entire genomic, not the entire genome. We're focusing on regions where the enhancer is, but it contains all the motif combination. And we're asking it to predict the data at very high accuracy without really knowing what kind of rules might be in the data. So so we don't have to put prior assumptions into our network. It simply learns the data from the bottom up. And if we can do so with very high accuracy, it has learned the sequence rule in a very general way, because we're testing on held out data. So it's sequences that it has never seen before. And we're now testing whether it can accurately predict the data. So if it has learned the data, of course, that's not helpful. We already have the data. But we can now go in and ask, what are the rules that the network learned? And that's very hard because traditionally neural networks are known as black boxes. And it's not so easy to extract the rules immediately because we learned this sort of in an unbiased black box kind of way. But the good news is that for genomics data, again, that's actually not that hard. We can open the black box and extract the rules. And at the very simplest level, we can use the trained network as an oracle. So we can hypothesize now, you know the network learned it, but now we want to understand the rules. So we can ask the network, what happens if we perturb the sequences? What happens to individual regions? Or we can also make this sort of more general. And so what happens when we always delete this motif? And so therefore we can extract the rules from the trained network. So a little bit more tedious, but it actually works quite well. And so the advantage, of course, is that we have the accuracy already. So we know it learned the rules really, really accurately if we have a really good model. So what we need is a good model, and we need interpretation tools to extract the rules. Okay, so once we have that, we really should be able to ask the question, is it important how these motifs for these transcription factors are arranged? And so this is a classical problem in transcription and has sort of been, kind of a good back and forth, because originally people assumed that there had to be syntax rules, so that the arrangement of the motifs was really important, because when you look structurally of how a transcription factor binds, it looked like there's a lot of interaction between transcription factors. It turns out they're not very tight, these interactions, so you can still question that. But overall, the idea has been from the beginning of the field that there should be a lot of syntax. Now, on the other extreme, there are people who came up with a model that now syntax does not matter. As long as you have the right combination of motifs, that's really all you need. So as long as the information display is together, it doesn't matter. So each information alone is sort of enough. So each motif would be one billboard essentially. And that's all you need as information. Now, so what I'm going to show you today, of course, the neural networks are ideal suited to solve this kind of problem. How do we look at combinations and syntax of motifs? So I'm going to tell you two stories. One is where we looked exactly that problem. So what are the syntax rules by which transcription factors cooperate with each other? And then the second part is now we can also apply similar kind of models to other data sets. And we specifically wanted to know what's the role of chromatin accessibility in gene regulation. Okay, so the first story is the syntax rules. And this is published, and this is really the story of how we got into deep learning to begin with. So I didn't start out, I started out as an experimental biologist, always kind of collaborating and doing computational analysis, but not specifically deep learning. But it turned out that really that was a solution to all our problems. So this is very nice to see. So I have always been interested in mapping transcription factor binding at the highest resolution possible, because I realized that in order to understand the code, I first of all need to be able to map motifs really precisely, because you can't really learn rules if you don't even know what the basic elements are. And we also found in the lab that transcription factor binding is not so simple that it simply only binds to its motifs, but the transcription factor by themselves cooperate with each other. They help each other bind. And so therefore, if we understand the patterns of binding, we automatically understand how they cooperate. And so if this is dependent on the arrangement, then we have a readout for syntax. So we developed in the lab a protocol called chip Nexus that maps transcription factors really at the highest resolution possible to date, which is a chip exo resolution, where there's an exonuclease that digests the DNA and then stops where the protein is cross-linked to DNA. And so what we're mapping are the stop bases. So if you're on the plus strand, it'll stop here on the left side of the protein. If you're on the minus strand, it will stop here. And so together, we're getting these footprints. So we're always displaying both strands in order to see these kind of footprints. And so one example, one postdoc in the lab did this for the pluripotency transcription factors in mouse embryonic stem cells, so OCT4, SOX2, NANOG, and KLF4. And so she generated chip Nexus data, and again, the data were super nice. So if we're taking a known OCT-SOX motif as a double motif, we can see both OCT4 and SOX2 bound. So each line is an individual region. And we can see a very stereotypical footprint of these two proteins. So that should allow us to identify where exactly these motifs bind. And we also found that the resolution is much higher than ChIP-seq, so that's shown in here. And also the specificity is higher. So for example, we look at OCT4 binding to the SOX2 motif, we can see that the reads accumulate as we do also in ChIP-seq, but we don't see these sort of spiky footprints. So that's very nice. But how do we extract this information? So this has been the big problem. So we know the information is good, but traditional methods don't allow us to extract the exact information that is. And that's just giving you a flavor of that. If you look at a very well-studied enhancer, it's also one of the nicest examples that we have. We see here OCT4, SOX2, NANOG, and KLF4 binding, and we see nice footprints, again, on the plus and the minus strand. And we see that there's overlapping binding, but what the rules were, and what exactly the sequences are that contributing to this binding is very, very hard to see by eye. And we don't even know how to exactly test this. And so this was then a very fortunate collaboration with Anshul Kondage's lab in Stanford, where we put our technology and our sort of focus on understanding mechanisms together with tools that they had developed to understand gene regulation using neural networks. And this was spearheaded by a graduate student from Julien Gagneur's lab in Germany, Giga Aussek, who did a really fantastic job. This was really, really a very, very fantastic collaboration. So, but I'm going to tell you everything about it. So we're able to predict really good models on this that are able to predict the high resolution data at the highest resolution itself. So first of all, we wanted the model to be highly accurate. I would say in general, that's a requirement. And in our case, we were able to train a model such that observed versus predicted data are as close as two biological replicates. That's really good, right? I mean, our data are great, and two biological replicates are really similar. Of course, the advantage is that at a sparser region, the biological replicates aren't doing so well, but neural networks are doing better. Anyway, so the models are really good. We also, of course, were able to train very large amounts of data. But we also realized that by predicting our data at base resolution, we're really leveraging the best readout possible. We're forcing the neural network to learn the data, the sequence rule at the maximum precision. So what Zika did is he predicts our data. This is the observed data for one transcription factor. And so he predicts that exact data for each base and both strands. And now he's doing this for all transcription factor and across many, 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 many regions. And so we're not just classifying like images usually, you classify an image and say, was this bound or not bound, but we're predicting the profile and also the total counts in this region. Okay, but of course the big challenge is how do we extract the sequence rules once we have a highly accurate model? How do we open the black box? Now the simplest way is to trace back the signal through the network to the input. In this case, we have 1 KB sequences. And the algorithm was already developed by Anshul's lab. It's a shot method, and it's called Deeplift, where we can trace the output back to the input And it gives us now weights on which of the bases contributed most to the prediction. So for an individual region, it looks like that. So this is just SOX2 and nanoc binding. And we immediately can see the bases that are important sort of form these motifs. This is in fact an OX-SOX motif. And we can see that SOX is dependent on it. But also nanoc, even though it doesn't bind this motif, really depends on this motif. Now we also see that this motif is really important for nanoc binding, and that in fact is the nanoc motif itself. And we see additional other motifs. Now this is one individual region. Of course, there are other interpretation tools required in order to see the patterns that are emerging from these sequences with very high important bases. And so once we do that, I'm going to cut a long story short, you can read up on that. We can then label these motif instances for each individual region, say, okay, now we know what the motifs are that are predictive for the binding of our data. Now, not only that, so now we have very precisely mapped motifs that are really leveraging the combinatorial learning of the network. Now we can also in silico mutate these motifs and go back to our model and say, okay, what do you think? If we delete this motif, what happens to nanoc binding, for example? And the beauty is that we're getting exact prediction for each individual region. So if we want to do an experiment on an individual region, it will make us very precise prediction what happens if we only mutate, let's say, 1 or 2 bases. So it's very powerful. Okay, so what do we see? So the question was, do we see rules of syntax? So there was the idea that it should be very strict syntax, or the idea of like, well, maybe there's none at all. And of course, people had already proposed that it's something in between, so we're not new to that. But what we can show is very precisely what the rules are. So first of all, there are these strict motifs. So 2 motifs that we already knew, like OCT4 and SOX2, that when together form these complexes. And these kind of strict motif, composite motifs are very nicely identified by BPNet. We're not going to talk about that today, but we found that to be very powerful also for other cell types. And on the other hand, we see relationship between transcription factors that we call a soft motif syntax. So in this case, it's not a strict requirement between 2 motifs, but rather that the binding cooperativity or the enhancement of binding that we see for a transcription factor depend on the distance to another motif. So we see that at closer distances, and in fact, specific distance it really likes, and further away, you still see an enhancement of binding, but it sort of decays with the distance. So it's sort of a distance function. And what we also saw, which was surprising to me, was that transcription factors don't always mutually cooperate. So in many cases, so we're comparing how transcription factor 1 depends on 2 and vice versa. And we see that 1 really depends on 2. So this is shown here, whereas the second one does not depend on 1. So this is what we call directional cooperativity. And that's really widespread. So since we published this, we looked in other cell types, and we really see this as a very common pattern. So just to give you examples of that. So this is when we compare 2 transcription factors at a time. And we see often that in one case, 1 is much more important to the other transcription factor. Now, you may also have noticed that here, this is nanoc binding, is very spiky. So what is that pattern? This turns out to be a 10 base pair periodicity. So that's, of course, the helical periodicity, so that the turn of the DNA. And we saw that already when we looked at the nanoc motif. So this is just a frequency. You don't really see much in the flanks, but we're looking at the motif. It's called the CWM. That is the network's version of the motif. And this says that, oh, if there's something AT in the flanks, nanoc binding is enhanced. So it contributes to the binding of nanoc. So here we see across many, many regions the importance of these bases. And so there's a very clear helical periodicity, which means that nanoc prefers to bind on one side of the DNA. So really suggests that now there's cooperativity, which we think now is protein mediated by protein-protein interactions, cooperative binding on one side of the DNA. And so when we do this, when we interpret the model, we don't just trust the first pattern that we see, but we go in with different interpretation tools or ways of extracting the rules to confirm this. And so we've done this many different ways. This is just one way. For example, we looked at here, not just the flanks, but we looked at the distance between different nanoc motifs. And we also see the preference for this helical periodicity. It was not dependent on the orientation, but on the distance. And also when we go back to the original data, so again, that's of course something we always want to see. If we learned the pattern, is it really there? We also see that in the original chip nexus data, we see this periodicity in nanoc binding. So this is really nice. And this is an example of how neural networks are really powerful in discovering patterns that you hadn't anticipated. So you didn't need to hypothesize this pattern, and they often sort of emerge by themselves. Yeah, so that is really great. And of course we can validate that. So we are a lab that does experiments, and so we can then go back and really validate that. And I'm not going to go show all of that. So we did CRISPR experiments to validate that directional cooperativity between nanoc and SOX2 in this case. So we took a region that has both a SOX2 motif and a nanoc motif, and we did CRISPRing for both the SOX2 motif and the nanoc. So here's a SOX2 motif. This is what BPNet predicts. It's the predicted profile wild type in the mutant. And then this is what we observe. So you can already appreciate that it's very similar. For an individual region, we're making very precise prediction at base resolution, and we're predicting exactly what happens when we mutate SOX2 motif. And in fact, we tested before we even generated the CRISPR mutations. We asked BPNet, okay, what happens if we do this? Will that eliminate binding? And BPNet said yes. And so we designed our experiments accordingly. So in this case, I'm not showing this, we're eliminating SOX2 binding, but we're asking what happens to nanoc? And we're seeing that nanoc binding is also reduced. So nanoc motif is here. And even though the SOX2 motif is mutated, we can see that nanoc binding is also now reduced. Now we're doing the opposite experiment where we're mutating now the nanoc motif, and we do abolish nanoc binding. We showed that. And we tested that before. But most importantly, we see that SOX2 does not care. And so we predicted that, and that's exactly what we see. So this shows that directional cooperativity between nanoc and SOX2. And I hope you can also appreciate there is a sort of long distance effect between nanoc and SOX2. Okay, so we showed really that this deep learning approach is very powerful. It's very powerful in learning the cis-regulatory rules, including syntax rules, and it's leveraging really the high resolution of genomics data. And we realized since then that it's also really good at even lower resolution data, such as ChIP-seq data, or even any other genomic data set. So because we're learning simply every base, it's not specific for the assay, and it leverages any resolution that you have. So that turned out to be really powerful. And that's why now Anshul's lab and also my lab, we're really applying this to all genomic data sets where it makes sense, where we know that there should be underlying sequence rules. And I'm going to talk a little bit in the second part of my talk about how we're doing that. And so what we see specifically in mouse embryonic stem cells, which were really well studied by many, many groups and analyzed, by genomics data sets and traditional methods, but yet we can go beyond that. And we see, for example, a helical periodicity for nanoc that was unanticipated, but is consistent, in fact, with some other previous factors in the literature for individual examples where this spacing seemed to be important. But we have not seen this in such a global genome-wide way, such as we see with nanoc. And we see this widespread directional cooperativity, which you might wonder, is that chromatin? And the short answer is yes, we definitely see that. So now I'm going to go to the second talk, unless you want to ask questions in between, which is totally fine by me, by the way. Any?
Yes. Hi, I've got a question. So,
yeah,
so presumably, well, not presumably, but this directional cooperativity, that would maybe suggest that, when one factor is dependent on the other, that probably the other factor may bind first.
Yes.
So have you sort of taken this a step further and looked at temporal binding? say during development and things like that? And also along those lines, can you somehow in your modeling predict along those lines protein concentrations?
Yeah, good questions, both. So the first one is interesting because I get that a lot, and I would always distinguish. So there are two scenarios. So binding first can mean either, even within a cell type, we know the transcription factor bind very transiently in the order of seconds. So even within a single cell type, you have lots of binding events. And so you could even say within one cell type that one transcription factor has to bind first before the other binds. So even under steady state conditions. And that I would say is also true, that yes, presumably one binds first, but it turns out that usually also is recapitulated in development. So it turns out that usually the transcription factors that are required first are typically also transcribed first, or in the embryo. Then the second story that I'm going to show you is also an example where that's the case. So we're looking in early Drosophila embryos, and Zelda is indeed the factor that comes first, and it's also the one that is required really to open up the regions for other factors to bind. So in this case, it really is both, that it's developmentally, it comes first, and it also is required first. But I would argue that, the developmental point of view is not always true, because development's much slower than what we're looking at in terms of transcription factor binding. As I said, one is an order of seconds. Usually development, is much longer time scales. And so it's very important to distinguish what exactly we're asking, you know what I mean?
Yeah.
And the second question was, of course, transcription factor concentration. And yes, that is super important. We know that transcription factor concentration drives the binding. In our models, we only learned it under one condition. So we are not changing the concentrations. But I know from other people's work, you can look at that. I mean, even like in the old days, you can look at that. And what you see is that when you have higher concentrations, you will access more lower affinity motifs. And that's indeed the case. But I do agree that it would be really nice to have data where we specifically manipulate only the concentration and then measure what happens. I think that's really, really important, because it could be that in part, the directional cooperativity is driven by concentrations. Yeah. So if one transcription vector is more abundant, it will drive the binding of the other one a little bit more. But I don't know how much can explain this directional cooperativity, but that's something we're really interested in.
Thanks.
Julia, I have a question about this as well. I wanted to ask this at the end of the presentation, but it's better to ask now. So in my mind, the cell type specific transcription program is made up by three really important components. So one is the DNA, the genetic code that you already taken into account. Mm-hmm. The second one, I think, is the chromatin that grant access to the DNA. Mm-hmm. Let's say we don't have to take that into account because the Yamanaka factors are pioneer factors, so they can open up chromatin. So they are strong enough to grant access to the DNA. Yeah. But the third one would be the molecular environment. All the RNA, non-coding RNA proteins present in the nucleus and the cytoplasm. Mm-hmm. That would contribute to that cell type specific gene expression. So one way of doing that would be to take into account the protein level of the 4 transcription factor that you are investigating. But if you could generalize this and somehow take into account at least the mRNA level of the whole transcriptome, maybe you could get the theory of everything, and you could predict all the cell type specific gene expression states. So would that be possible to use the transcriptome as an input as well?
You could. I mean, you could put anything to give anything to neural network and see, but that would only be if you wanted to. So the thing is, though, the network is only trained on this one cell type, right? Yes. And so whenever it's given, it doesn't know that, right? It just learns the rule for this cell type. And so, of course, I think what you're getting at is, well, what if you learn it for different cell types? And is there information you're missing? But I would argue that if it's encoded in DNA sequence, it should be able to learn it. It doesn't really need the additional information. At least my first attempt would be to try to learn it from DNA sequence. And by the way, this is also the answer for the chromatin accessibility. And I'm going to show you that actually, this is the second story, of course, that chromatin accessibility, even though people use it as a prior, as something that should be given, it's also encoded by the sequence, and it can be learned, in fact. So if it can learn by sequence, then ideally that's better than giving it a lot of prior. So the reason is, if you give it as a prior, then you don't force the network to learn this information. So people have done that quite a bit as sort of a way of like, well, if I don't know that cell type and I have no way of learning the precise rules, I'll just give it the chromatin accessibility. And that contains a lot of information. And that, of course, helps the prediction. But it also means that you haven't really learned the rules from scratch. So the way I see it, that as long there isn't that much that you can't learn from sequence. Well, there probably is, but we haven't. That's not where we started. Basically, we started with the things that we think are encoded in the DNA sequence. And in terms of environment, so the thing that we're specifically looking at, I mean, obviously RNA is not necessarily our focus, but we have looked at enhancer RNA, and we're very interested in that. But the most obvious thing to us is the signaling. So signaling is a very strong determinant of enhancer activity, in addition to the transcription factors. Or, I mean, of course, there are transcription factors that are downstream of signaling, and they have quite a strong impact on activity. And that's a story that I'm not presenting today. We have looked at, and indeed it is the case. The signaling factors are the ones that come in last, and they're dependent on the cooperativity of the other transcription factors, but they bring in activity to the enhancer. And the signaling pathways are really what's sensing the environment of the cell, and often also the state. And so that's a really a way of translating that directly to the level of enhanced activity. So that sort of is very straightforward, as something I'm traditionally have studied for a long time. And so I'm very inclined to look at that. But you're right. I mean, you could also do, I mean, there are people who are using, let's say, any kind of information like single cell RNA-seq data. Can we use that for a neural network to make predictions? So you're sort of taking a more black box approach to this and say, hey, give me anything that I can to predict what the cell's going to do, right? And so people have done that, of course. But it's not specifically getting at the question of, these sort of fine-grained rules of how transcription factor function. So I sort of envision that will sort of converge later with what we're doing. So I'm not saying that any approach is not useful. It's just kind of a different question. And at the moment, we can't really converge that very easily. So it's also a strategic question of like, what do we learn first? What do we focus on first in order to learn general rules? And how do we, I mean, the cell type specificity is really the big problem, right? We can do this really well right now for individual cell types and different data sets, but how can we build more general models? This is the big challenge. And I think you're getting at that. Like, do we need to give it more information? And we can talk more about that. I could talk about forever. So
that would be great. That would be great. Maybe after the meeting we can have a separate conversation, because I still have some questions. You convinced me about this. So I have other questions as well.
Okay, cool. Yeah, so I'm going to tell you about the chromatin accessibility. You actually gave a perfect segue into that. So the role of chromatin accessibility is often viewed differently. And you had one view that I did represent is sort of that, oh, well, that should be a prior, right? That can be developmental history, something that the cell did previously, so we don't really know about it, so we need to know it, right? The other take would be that it's, and you mentioned that too, that the Yamanaka factors, many of them at least, are pioneer transcription, OCT4, SOX2, KALA4, pioneer transcription factors. So really, accessibility should be the readout of the consequence of pioneer transcription factor. And the third possibility is, and something that I also see quite a bit in talks, is that you simply use the open regions to find all the motifs that are relevant for your cell types. And that seems to work. I mean, you seem to discover lots of motifs. So what is it really? Now, the problem with this is that normally it's very hard to distinguish between cause and effect. So that's what I call the chicken and egg question. So is when we see a motif in a region, so we take all the regions that are open, and ask what are the bound transcription factor motifs that are enriched. And we're looking for, of course, for statistical enrichment. But we don't know whether the motifs are the ones that make these regions accessible, or whether the motifs are those that are found there that eventually help the enhancer to become active. And so I hope you can appreciate intuitively that a neural network should be much better at learning the causative relationship between the motifs, their spacing and the combination, and the accessibility, because we're learning it for each individual region. So we have the exact combination and spacing of motifs, and we now have to predict each region and its accessibility. And so by eye, you could see already that this motif presumably is more important for, it is more important to predict the accessibility accurately than other motifs. And this is exactly how BPNet works. And so we wanted to apply this approach to accessibility and see what we can extract from that. And so this was a project spearheaded by Kaylin in the lab, a graduate student who did all the experiments and some of the analysis, and was really greatly helped by my bioinformatician in the lab, Melanie, who already was on the BPNet paper and really is an expert on BPNet. And so we also wanted to show that we can apply it to Drosophila embryos. It's not always the same, genome is smaller and all of that, but we were able to apply this. And we specifically like the early Drosophila embryos because we are establishing now chromatin accessibility de novo. And we can also look now over time. So come back to the question, we now have a time course of accessibility that we can look at. And so we now want to predict, of course, the data using BPNet. And we also wanted to look at binding, since we had done this successfully before. And we wanted to make sure that we're really learning the motifs of the transcription factor very accurately. And so I cut a long story short. So we did the same approach that we did previously, where we took 6 transcription factors that are known. So Zelda here is the pioneer that comes first and is known to open chromatin. We worked on this before. There's also Gaga factor, who's opening chromatin. And then these are patterning transcription factor. Dorsal and twist, we worked on before, dorsal ventral patterning. And sure enough, the model learned the known motifs, and it also learned the relationship between them. So it learned that Zelda was really important. So this is the motif itself, and this is importance for the binding of these transcription factors. And we can see that Zelda is important for the binding of all the transcription factors. Gaga, interestingly, even though it's supposed to open chromatin, we do not see that it's important for the binding of the other transcription factors. So this is all consistent. So overall, we did discover no motifs. We also, when we look at individual enhancers, we can see that it matches what people had mapped, painstakingly before, very easily. And we see that hierarchical relationship between Zelda and the other transcription factors. Okay, so now we wanted to look at chromatin accessibility. And so now Kaylin performed a time course of chromatin accessibility over time. And we see that chromatin accessibility increases as expected. Now, the problem is we wanted to leverage the base resolution of BPNet. So we want to learn the data at base resolution like we did before. But ATAC is experimentally biased. So it's a transposon TN5, and it likes to insert at sites where it sees certain sequences. So normally you don't really see that because you map the entire fragment. And so then you see something like that. But we wanted to leverage the resolution, really map where TN5 inserts itself into DNA, because that's where the accessible chromatin is. But when you do that, you see a very spiky profile. And so Anshul's lab developed a really clever method to get rid of this bias. So what they do is they train 2 models, one on the bias, they train that first, and then they train on top of that a second model. And so now we're training on predicting this exact data, but we can now take that bias model away, and now we only have the model with the biology, and this is what it predicts. So we can now predict what the chromatin accessibility should look like if the TN5 bias wasn't there. And so this worked really well, to cut a long story short. And so when we now go in and ask, so this is one enhancer region that's well studied. And we ask, okay, so it predicts now this accessibility. What's important for the binding? And we can already see that these spaces stand out as being very important. We have, of course, mapped the motifs already. And we can see that indeed the Zelda motifs are what's driving the accessibility in this region. But we also see that dorsal, for example, also seem to have a contribution to the accessibility. So it seemed to be a little bit more complex than simply Zelda opening up these regions. And so we set out to sort of analyze in more detail of how this occurs. Okay, so first of all, we're looking at how important are the motifs for accessibility in general. So as you saw in the individual example, Zelda is very important, and that's what we expected across the time course. Gaga factor, which we thought was opening chromatin, indeed in this model, it does really open chromatin. And then we see a contribution of the other transcription factor, but just a little bit. So that raises the question, what are the rules that it learned? So what we did now in order to analyze, so we wanted to combine binding versus accessibility. And in order to do this, we used the motifs that we learned through binding because we're more confident in those. With accessibility, there are also other transcription factors that learns, the motifs get a little bit fuzzy. And so we took the motifs from the binding model, but now we can ask exactly the same motif that's bound by factor, how does it contribute to accessibility? And this is shown in here. And I hope you can see, so this is the accessibility counts contribution. And this is the binding counts contribution. So counts is just the overall signal that we get in the region. And what we can see is that there's quite a good linear relationship between binding and accessibility for Zelda and Gaga factors, so the pioneer transcription factors. Now you may say, is that trivial? Well, yeah, of course. I mean, and you also see that there is a dependency on motif, right? So here, the more yellow is higher affinity, and the more blue is low affinity. And so when we see high affinity, we see more binding, and we see more accessibility. Very consistent. Well, it's actually not trivial at all. So to me, that was surprising because pioneer transcription factors, the definition is that they bind in the presence of nucleosomes. So the idea is they bind outside where they're structurally constrained in their binding. And in fact, previous studies suggested that they bind partially to their motifs, or in vitro, they often bind only at the diet or only at the linker regions. So it's very, very constrained. But what we're seeing here is that essentially the motif, once it's there, it mediates accessibility. So that's what the model tells us. So to me, that was surprising that it seemed to like, okay, well, as soon as the motif is there, Zelda will open up the region. And so we wanted to test that. And so first thing is we tested whether the model learned motif affinities correctly. And this is very nice to see that it did. So we used a protein binding array in collaboration with Tim Hughes and Chris Rushlow. And here is just the binding signal on the microarray. And then here is what the BPNet model derived. And this is just a statistical significance. So we can see that that matches really nicely. And that also matches with previous like EMSA's result, of course. And so we're very confident in identifying high affinity motifs versus, for example, this low affinity motif. So now we can go in and ask experimentally, what happens if we deplete Zelda? What happens to high versus low affinity regions? And so this is what we did in Zelda minus embryos, which we validated. And this is just one region as an example with a high affinity motif. Over time, you can see that there is really a big difference between wild type and the Zelda mutant, and it increases over time. And we see the same thing for a low affinity motif, where over time, I mean, the effect is smaller, but we're still seeing quite a bit of an effect over time. Whereas for Gaga, of course, the control, we don't see a difference. When we look at this now across many regions to get confidence, we can see indeed that the low affinity motif does drive accessibility, but to a lower extent than the high affinity. So I thought that was really cool because it does show that pioneering is very, very precise. And it also gives us sort of a definition of pioneering, because there seemed to be a really good relationship between binding and opening chromatin. Okay, so now the question is, what about the other motifs? And so here we use the data that we generated for Zelda, so the accessibility wild-type versus Zelda embryos, we leveraged that to ask, what about the other motifs? So here we're looking at the loss of accessibility in Zelda mutants. So here we're looking and I should say the previous, we're only with the Zelda motif. So we're eliminating the influence of other motifs. And this is what we see. We see a very consistent drop of accessibility if there's only a Zelda motif. But now if we're looking at other motif combinations, those are all BPNet map motifs. So we know that they result in binding, but we also know that the binding is dependent on Zelda. And so that's why now we're seeing, but dependent on the motif combination, we're seeing a much bigger loss of accessibility. So this shows that Zelda is not acting alone, that the other motifs, when combined with Zelda, because they need Zelda for binding, they also cause a loss of accessibility. So that is interesting. So this is also what the BPNet model previously saw, and we can show this by mutating. Again, we're giving it in silico sequences where we mutate a motif and ask what happens here. So this is the enhancer that I showed you before. And we can see, well, Zelda has a huge effect. So red is the effect, the difference between wild type and mutant. And it's a big difference. But when we mutate a dorsal or a caudal motif, we can also see, especially when here, where these transcription factors are active, there also is a loss of accessibility when we mutate the motif. So those motifs are also contributing to accessibility. That's what the model predicts. And that's what we're seeing in here in the Zelda mutant. So the question is how? And so to do this, we took we looked at dorsal, because dorsal is something that we studied before, and we knew that dorsal functions both as an activator and a repressor. And so what was interesting, so in this case, we used a mutant where dorsal is no longer active, and we looked at how all the genes change in accessibility. And the striking part is that when we look at genes that are repressed by dorsal, which are these genes, they actually go up in accessibility, whereas genes that are activated by dorsal go down in accessibility. So this suggests that it's not simply dorsal binding that generates accessibility, but it's the effect that it has on enhancer activity that determines whether the accessibility goes up or down. And we wanted to validate this. So this was really cool because we studied this before, and we knew that dorsal requires Capicua. So this is a repressor, and they're repressors. So in these sites, you have, in these repressed binding regions, you have Capicua binding motifs. And so we knew that if we look in this mutant, we're simply abolishing the ability of dorsal to repress. So now we're predicting that only the dorsal repressed regions should be affected. And so there's the yellow ones, and indeed, they still go up. They are affected in the Capicua mutant. Whereas the ones that are activated by dorsal, those should not be affected. And sure enough, they are the same. So this really does suggest that accessibility is coupled to enhancer activation. And so to cut that, so I mean, we validated in different ways, but to just show the model of how this works. So I showed you, first of all, that pioneering by Zelda and also by Gaga factor is very consistent. So whenever you have the motif, you have a contribution to accessibility. And we know that those factors function upstream in terms of binding. So they have to come first. And they are actually indeed developmentally usually also present first. So that was surprising because these transcription factors usually bind outside the nucleosome. So this was surprising that they so consistently contribute. Now, in contrast, these patterning transcription factors, it really depends on the context, whether they contribute to accessibility. So they do depend on cooperativity with the other motifs. And so I showed you here this sort of hub model, the condensate model, not because it's fashionable, but in this case, we really had very strong data from imaging to suggest that these transcription factors do indeed form hubs that are dependent on ZELDA. So it's very consistent with the model that ZELDA comes in, open these regions, and these transcription factors then come and bind and form hubs. So presumably in this model, these hubs form in a DNA-seeded kind of fashion, dependent on multiple motifs here. So this is currently certainly my favorite motifs of how these transcription factors are dependent on this sort of soft motif syntax. Okay, so this is what we learned from that. So if we go back to our original question, was like, okay, well, what is it? Is this just developmental history that we're looking at? It's like, well, yes, we actually did see an effect over time, and we don't know exactly why that is, but we know that it was not required. We didn't need previous time points to learn this information. We could predict accessibility from sequence alone for each time point, which is nice. And so the idea is that accessibility is indeed sequence encoded. Now, second was like, is it the readout of transcription factors? And the answer is yes, we definitely see that. And it has the most consistent effect. So ZELDA and GAGA factor, but we also see an effect of enhanced activation. And this explains why we also see other motifs there that can be discovered using accessibility. So some of them do contribute if they contribute to activation, essentially. So that means you're not necessarily learning all motifs. Not all motifs necessarily that are found in this region are contributing to the accessibility. So repressors, for example, do not contribute to accessibility, not positively at least. But strong activators likely also will contribute to accessibility. Okay. So with that, so what we're now pursuing with this deep learning approach is to learn different genomic data sets and how sequence rules contribute to how, what their readout is. So we're coming from transcription factor binding. We are looking at enhancer transcription, by the way. I just didn't have time to talk about that. Well, as also mentioned in the introduction, we have studied Pol2 quite a bit, and so it was natural to us to look at enhancer transcription by Pol2. So it's very interesting to us. We're looking at histomodifications, and of course, long term, it's very important to understand what the target genes are. To eventually have really comprehensive models of gene regulation. Biggest challenge, of course, is to put this all together and also have something that's generalizable. So currently what we're doing is using deep learning combined with experiments to really derive rules with the goal of matching these rules to known biology. So in the case we can say our directional cooperativity, there may be chromatin accessibility, or we've seen motif affinities we can extract from the model. So we're seeing biophysical constraints. And so we're really extending that to other questions. So overall, the goal is to use this knowledge to eventually be able to have more broader predictive frameworks that might be able to predict gene regulation in cell types where we haven't gotten that much information. Of course, how do we get there? I think there are a lot of pieces that we need to put together. So we're really focused on tying it back to mechanisms. Of course, there could be computational innovations that would also really help, or large-scale data sets, of course, is also tremendously helpful. Yeah, but this is the approach that we are taking with this. So what I'm really advocating is that it's completely changing the way for biologists, the way we do experiments and the way we study things, that really should us provide with a sequence basis for biology. And I sort of compare this to the progress in navigation in human mankind. So initially, we were, local guideposts were all that people had. And so that's also in the old days, experiments were done based on educated guesses of what genes or proteins might be important. Now, once we had genome-wide data, we could use that information. So maps were obviously tremendously helpful for a long, long time. But now that we have small computers in our pockets, it's really amazing that we don't even need to do a lot of work. We can simply use our device and ask, okay, what time are we going to arrive? How do I get there? And so I would argue that biology will undergo the same transformation where we can use neural networks to make very accurate predictions. And so therefore, it allows us to make much more focused experiments, so predict beforehand and let us test our favorite hypothesis rather than doing everything in large scale. So this, I think, is really exciting. And so obviously doesn't eliminate experiments, but I think it's sort of a really nice interplay between the deep learning and experiments that I really enjoy. So with that, I wanted to thank, of course, all the people in the lab who've been tremendous in sort of embracing this new technology and see what we can do with that. And so with that, I'll take more questions.
Thank you very much for the great presentation. Questions from the audience?
Well, I can.
We have a question. Yep.
Yeah, great talk. Yep. So I guess there's one question, two parts. I'm glad in the second part you mentioned the affinity of transcription factors, which is obviously really important, and sort of linking that to the first part. And I can't remember the speakers, unfortunately, but at an EMBO meeting last year, she was looking at limb development and enhancers involved there. And what sort of blew me away was that she sort of predicted affinities of transcription factors to the enhancers. And it was actually involved that the affinities were not that high. So, for example, if one was a maximum, they usually range between, say, 0.3, 0.5. And what she found that if she mutated the enhancers and actually made a stronger transcription factor binding, it actually inhibited development of limbs. So I'm wondering, with the affinities, going back to your prediction, maybe you can actually predict the relative affinities of the SOCs and all the other transcription factors, and get a better understanding of the affinity roles in their function. So it's not just a matter of binding or not binding.
Absolutely. So I didn't show that, but of course we can do the same thing, the affinities for the OCT4 and SOCs too. So we definitely have that information. We have not analyzed it to that. That is something we're planning to do. I've done a little bit actually, but not in a yet sort of story ready version. But we do have another story where we looked in trophoblast stem cells. And there we saw something really interesting. So there is a double motif. This is of TEAT4, so hippo signaling. And we found that, so first of all, it binds to a TEAT4 motif, just a classical motif, but there's also a double motif. And this double motif is highly cooperative, and it's usually present in the genome as low affinity. And so we can't prove that, but I think the assumption is that it has been selected because otherwise it would be too high of an affinity. So the TEAT4 either binds a single motif or really low affinity double motif.
That's interesting.
And so this, of course, begs the question of like, well, there has to be a price. Like if it's too high, something is either you can't regulate it or I think there are different explanations of why that is. And I have seen also a talk where, for example, the same motif is bound by the activator and a repressor, and so it could be the interplay between those two where a high affinity might not be good. In other cases, a low affinity might be preferred because you want to make sure that it responds to only high concentrations of the transcription factor. I kind of, for a second, I thought that you were going there. So if you have a morphogen.
No, actually,
that was probably the original question, but then it turned out it was different, right?
Different, yeah.
Exactly.
So evolutionary, it actually regulated the DNA sequences to make sure it's not too strong.
Yeah, well, see, it can't. So for the cases that we've looked, it wasn't the case. So we have very high affinity. It's not generally true that a high affinity is not good, but there are cases, as I said, that double motif where we're suspecting that high affinity is not necessarily good. But there are many other reasons. That's what I wanted to say for having a low affinity motif. So yeah, absolutely. I think this is the great advantage of the neural networks, that we can identify low affinity motifs. So usually if you scan for low affinity motifs, you have no confidence that this is really indeed a low affinity motif that's being bound in vivo. So we have that confidence. And so now we can really go in and ask those questions. Yeah.
So if I may be selfish and quickly ask another one about chromatin accessibility. Of course, a big factor of accessibility is nucleosome positioning, right?
Yeah.
And so inherently, some nucleosomes are positioned in enhancers that allow transcription factor binding sites to be accessible or covered, right? But those that are made accessible are inherently more accessible. Yeah. So I'm wondering, I wonder if it hasn't been already done, but you know, there's lots of high resolution nucleosome maps out there, certainly in vitro. So I wonder if it'd be wonderful if you could train and then predict nucleosome positionings, right? And that you'd lay that over your accessibility, and because then you'll find, like I said, those are sites that are inherently accessible because the nucleosomes are positioned away from those binding sites.
This is exactly what we're doing. In fact, we've been doing it for already quite a while. It's not easy. So the chemical mapping data, they exist in mouse embryonic stem cells. So obviously we have analyzed those. We do see a preference of the OXOX motif to be on a nucleosome, in fact. Yeah. So suggesting it's actually removing the nucleosome. It's inconsistent with the in vitro data, by the way.
Yeah.
We also have trained models on nucleosome occupancy. So what you're referring to, I think, is at least the yeast 4-5 model, where it's very well documented that the motifs that is between the nucleosome is the ones that drives the initial binding, and then once the region becomes accessible, it can also bind to the occluded region. So we're also looking at that. And we have a nucleosome model for yeast. We've done a little bit interpretation, but it's not ready yet. So this is definitely a long-standing question, and we're pursuing it really seriously. But I can only tell you just a little bit about it. So we do see, for example, so we knew that there is AT frequency in the nucleosome positioning, and we can see that quite nicely. Yeah. We also, there is, of course, the idea that the dinucleotide frequency of either AT or CG is important. We see that it's probably a little bit more complicated than that. So it's not just any AC, any GC or AT. So there seem to be a much more specific code. So I suspect it has to do with DNA bending, because there's loop-seq data, if you heard of that. And so that also suggests that, but we have not put that together in any coherent way. So we really wanted to make sure we're correcting for biases in the data. And so it ended up taking a lot longer than we wanted. And so we don't want to put anything out there that's too preliminary. So this is a hard problem, that's in other words, but it's super interesting. And I totally agree with you. It's an obvious question. And
thank you.
Thank you very much. Tomasz has the next question.
Thanks for the interesting talk. And I would like to ask a little bit about, in your models, you can clearly see that, for example, non-OG or SOX2 binding, you have the well-known sequence, which is the binding sequence. But in your model, I also see that then there are all these sequences around, which is much lower important, but they are still coming up there.
Yeah.
So have you tried, okay, so just one step, but I try to explain where the question is coming from. So we know already for a long time that if you just have a motif and you just look at that in the genome, there are many motifs which are not occupied by the transcription factor, and then many others occupied which are not really good motifs, but still quite good. So you think this is all explained by just simply this codependence of the different transcription factors, or all these kind of low, so these bases which are just really kind of low predicting values around, do they have actually a real function? I don't know, does it make sense what I'm asking? I don't know if I could really...
No, it makes complete sense. It makes complete sense. I was just, sorry, I was a little distracted. I looked at the chat, but it's not related to your question. Yes. So I should say that we're not predicting it in the entire genome, right? So we're selecting regions that are bound by transcription factors. Now that includes regions that are not bound at very high levels at all. But we're not using regions that are heterochromatin regions, for example. So I bet you can take region, and for example, if you put it in a star-seq assay or any reporter assay, you take regions out of heterochromatin and you put it into a reporter, you can drive expression. So that shows that sequences that are even capable of driving enhanced activity may not necessarily show that in vivo. So there are definitely additional layers that we sort of specifically avoided, intentionally avoided. So we're only really looking at regions that have the capability of becoming bound. And so within that limit, the model is really good at predicting that and suggests that within that framework, it really is the combinatorial binding that's driving quite a bit of that. So if there's no pioneer factor that opens the region, then the other transcription factors bind at very, very low levels. Now, it doesn't bind nothing. I think this is the good part, that the model is so quantitative that it's not asking, like, is this bound or not bound? It just asks how much. And so if something is not bound, it just predicts it tiny, 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 essentially. That's sort of the trick.
But essentially, if you would try to let's say you would still keep the sequence, so you would do this, you would use now reverse your model, or you give another sequence and try to predict whether you would have binding, and you would still keep the binding motif, so that 6 nucleotide binding motif for, let's say, for both transcription factor, but you would completely change this almost randomly the sequence around.
Yeah.
Would it still, have you ever tried, would it still predict to have binding, or it would not? Because of course these bases around individually, they had a low kind of importance, but altogether, do they have actually, so for example, is it possible that RNA or something else, they are still important to find the right binding motifs, or really only these six nucleotides, which is just the DNA protein binding, and that explains everything? Or are these surrounding bases actually important? Did you ever try that?
Yeah, it's a really good question. And the short answer is yes and no. So what we have done is sort of we isolated motifs and asked, so we give it randomized sequences and ask what's the binding. So I've looked at that quite extensively, and so we can validate this sort of binding cooperativity rules in this kind of randomized background. But what you're asking is actually kind of different. You're asking, how much are we missing? Yeah. And that's a good question. We have not pursued that to the extent that we could, perhaps. So you're right that we're seeing this sort of fuzzy, low, small contribution. And so some of that are low affinity motifs. So in our initial paper, we were fairly stringent and we didn't call low affinity motifs. And we did realize that. So I think we sort of estimated we mapped about 40% of the motifs only in that paper. Since then, now we have tools we're more confident in actually identifying low affinity motifs. So the problem is, of course, you still have to know what factor binds if it's low affinity, even though it's important, but what does it do? What does it bind? So we're getting better at that. But I probably would think that there are still things that we don't fully understand and that may not even be real. So we don't know, is the model sort of trying to cheat and look at CG, dinucleotides and sort of using that as an indicator of some sorts that may not even biologically relevant, right? So that we cannot exclude that sort of the rest is either biology that's super interesting, that's coming from nucleosomes and that we haven't learned yet, or that it's sort of noise of the model and just trying to learn information that's not really there, or low affinity, which we have a little bit more access to and kind of getting at eventually. So I do agree that that is still a little bit of an unknown. We haven't specifically tackled that question, but we have that sort of at the back of our mind. And sort of slowly, yes, by comparing different models. And so we'll get a feeling for how much is there that we're missing or by looking at nucleosomes. But yeah, I mean, this is sort of a little bit of a limitation of the black box model also, that yes, you can see, even if the predictions are, super good, whether the rules that you extracted are everything that the model learned is no, you don't really know that, right? Unless you build it from the scratch up, like kind of you're suggesting, which would be like, hey, we're taking all the motifs that we learned and all the rules, and now we're building it back up. and say, how much does that predict?
Yeah.
So that we have not done, so reverse engineering. I think it's a really good question. Yeah, we have not done that.
Thank you.
Many thanks for the answer and all the great questions. So if you have no further question, we will wrap up here. And many thanks to Julia for making the time and giving us this excellent presentation.
Yeah, well, thank you. Nice to meet you.